bringing together of male and female protoplasts or nuclei, that is plasmogamy, is achieved by planogametic copulation, gametangial contact, and gametangial copulation. Now we will discuss them briefly one by one. Number first, planogametic copulation. This type of sexual reproduction involves the fusion of two naked gametes where one or both of them are motile. The most primitive fungi produce isogamous planogametes. Example: Syncytium, Olpidium, Plasmodiophora, etc. The anisogamous planogametes are only found in the genus Allomyces of order Blastocladiales. Monoblepharis illustrates the most advanced type of planogametic copulation. It produces small opisthokonte male gametes in large numbers inside the sex organ called antheridium. The oogonium produces a single large immobile female gamete, the egg or ovum. The male gamete enters the oogonium and fertilizes the egg. Number second is gametangial contact. Here, a group of planogametic lover fungi, that is Oomycetes, produce non-motile gametes that are never released. The male gametangium is called antheridium and the female oogonium. No sperms are organized. The gametangia do not actually fuse and they never lose their identity. The antheridium puts out a slender tubular outgrowth, the fertilization tube, at the point of contact with the oogonium. Meiosis is gametangial. Examples are Saprolegnia, Achila, Pythium, Phytophthora, etc. Then we have the gametangial copulation. In another group of planogametic lover fungi, that is Mucor, Rhizopus, etc., gametic union is brought about by the fusion between the gametangia. The uniting gametangia lose their identity in the sexual act. The intervening walls between the two gametangia dissolve, forming a common fusion cell. Plasmogamy is immediately followed by karyogamy. The diploid contents of the zygospore or the diploid nuclei undergo zygotic meiosis. The meiospores on liberation germinate to produce a new haplomycelium. The ascomycetes are characterized by the presence of an ascus. The ascus is the product of sexual act and the ascospores produced inside it are thus sexual spores. The young ascus is always binucleate. These two nuclei undergo karyogamy to form the diploid zygote nucleus. This is followed by three successive nuclear divisions. The first division is normally reductional, the others equational, forming usually 8 ascospores by free cell formation. In the family Saccharomycetaceae, however, modification of the above process can be seen as complete suppression of the specialized gametangia and copulation takes place between vegetative cells, that is somatogamy. The gradual suppression of the sexual phenomenon is characteristic of yeast fungi. The formation of ascospores in yeasts is contingent upon a sexual process. Of course, no sexual organs like antheridia and oogonia are produced. Instead, the sexual process is extremely simplified, however, occurs through plasmogamy, karyogamy, and meiosis. In Saccharomycodes ludwigi, the ascospores directly function as gametangia and copulate. Sexual fusion takes place within the ascus between the two adjacent ascospores of opposite mating types, that is plus and minus strains. In fission yeast, Schizosaccharomyces octosporus, each haploid somatic cell is a potential gametangium. At the time of sexual reproduction, two somatic cells come to lie side by side and each sends out a short narrow beak-like process. The two meet and the intervening walls dissolve to form a conjugation tube, the latter functioning directly as ascus mother cell. This process, though similar to the oogamous type, in its initial stages, has many evolutionary and distinct features. In this method, well-developed uninucleate or multinucleate gametangia develop. The males are designated antheridia and the females ascogonia, which are provided with a specialized receptive hyphae, the trichogyne, not formed in the oomycetes. The male nucleus or nuclei that migrate into the female via the trichogyne do not fuse but remain associated, thus undergoing dikaryophase. Then one or more ascogenous hyphae arise from the ascogonium, and ultimately SI are formed by the crozier or hook method. This type of sexuality occurs commonly in taxa belonging to several groups such as Planogametic, Pyrenomycetes, and Discomycetes. Most species of Aspergillus are only known to reproduce asexually. The sexual or perfect stage is rather rare. Possibly the species lacking it have lost it during the course of evolution. In some, there is a degeneration of male nuclei, whereas in others the antheridium is rudimentary, non-functional, or even absent. In Aspergillus herbariorum, the tip of the antheridium fuses with that of trichogyne, but there is no migration of contents of the antheridium towards the trichogyne. In other species like Aspergillus nidulans, the antheridium may develop late. By the time the ascogenous hyphae have already been developed from the ascogonium. In a few species, the male nuclei in the antheridium degenerate before the antheridium has reached maturity. The antheridium in some species such as Aspergillus flavus, Aspergillus fischeri, Aspergillus fumigatus does not develop at all. Only the ascogonia are seen to develop. In Penicillium also, ascocarp formation is absent. Only a few species are known to reproduce sexually, for example, Penicillium vermiculatum and Penicillium stipitatum. Here, the mycelium contains uninucleate cells from which develops a uninucleate ascogonium. The ascogonium elongates and its nucleus divides several times to produce as many as 64 nuclei. Simultaneously, a uninucleate antheridium also develops and coils spirally around the multinucleate ascogonium. The antheridial nucleus does not migrate into the ascogonium. It remains very much there in the antheridium even after the development of ascogenous hyphae. This indicates that antheridia are formed but remain non-functional. Some Discomycetes like Morchella show the total absence of both antheridia and ascogonia. The accessory parts of sexual process are completely suppressed. Consequently, the sexual process is extremely simplified. Plasmogamy in Morchella takes place by the following two methods. Number first, somatogamous copulation. Here, two vegetative hyphae of the subhymeneum region of the pileus come in contact. The intervening walls between the copulating cells dissolve at the point of contact. Later, the two multinucleate protoplasts intermingle in the fusion cell. The two functional nuclei, one from each copulating cell, then form a dikaryon. All other nuclei in the fusion cell disappear. Later, the ascogenous hyphae arise from the fusion cell containing the dikaryon. Each young ascogenous hypha receives a pair of nuclei. These are the derivatives of the parent dikaryon. The ascogenous hyphae afterwards become septate. The terminal cells of these hyphae function as ascus mother cells. Number second is autogamous pairing. In Morchella elata, any vegetative cell of the subhymeneum may establish a dikaryon. It is accomplished by pairing of two of its own vegetative nuclei, hence called autogamous pairing. The remaining nuclei in the cell degenerate. The cells with dicaryons develop ascogenous hyphae as in somatogamous copulation. The two nuclei in the ascus mother cell undergo karyogamy, forming young ascus without crozier formation. This is followed by meiosis and 8 ascospores are formed. This is the final stage in the elimination of both the antheridium and ascogonium. The sexual process becomes extremely simplified. It takes place by the copulation between the cells of two compatible hyphae at any point along the mycelium through anastomosis. Two strains of the opposite sex, however, are necessary. Finally, there is disappearance of even this impulse towards sexuality. The two vegetative nuclei of adjacent cells of the same homothallic mycelium come together to establish a dikaryon. The two nuclei of the dikaryon fuse in the ascus mother cell to form a synkaryon. The later, by meiosis followed by mitosis, gives rise to 4 or 8 haploid ascospores within the ascus. Total apomixis, that is absence of sex, has been reported in some cases. In Ascobolus equinus, although solitary ascogonia are produced, karyogamy is totally absent. Only haploid nuclei are found everywhere. Only a single haploid phase occurs in the life cycle in which all nuclei are alike. This is indeed a rare and exceptional phenomenon. In Basidiomycetes, plasmogamy takes place without karyogamy. The spatially differentiated gametes and gametangia are usually absent in the Basidiomycetes, excepting rusts. But there are other ways of coming together of the two compatible nuclei. They include: number first, somatogamy or mycelial fusions; number second, spermatization or oidization; and number third, fusion between two oidea, which are small hyaline one-celled and uninucleate spores formed on oidophore tip. Thus, sexuality is said to be reduced in Basidiomycetes. The primary manifestations of sexuality, that is, the union of cells and nuclei, are obviously present in the Basidiomycetes, in spite of the absence of such secondary criteria as specialized gametes and gametangia. The compatibility mechanism, in fact, is much more highly developed in Basidiomycetes than the other fungi. Let us discuss the sexuality in Astylago and Agaricus to illustrate sexual mechanism in this group. No sex organs are developed in Astylago. The sexual process is represented by 3 fundamental phenomena characteristic of it, namely plasmogamy, karyogamy, and meiosis. In Agaricus and other mushrooms, the sexual apparatus in the form of sex organs is completely lacking. Their function has been taken over by the somatic hyphae, which are heterothallic. The fusion between two somatic hyphae of plus and minus strains represents the first stage. Plasmogamy is accomplished by somatogamy or somatogamous copulation. Fungi imperfecti or Deuteromycetes comprise of a group of fungi in which only the asexual or imperfect stage is known. The sexual stage, also called the perfect stage, is completely absent. The sexual spores, like sporangiospores, ascospores, and basidiospores, are either non-existent or have not been observed or discovered so far. Reproduction takes place chiefly by the formation of exogenously developed asexual spores called conidia.